Howdy, my name is Samuel Francis and I'm a rising junior attending West Virginia University working in neuroscience research. The research I'm about to present to you was done with the assistance of graduate students Oliver Cook and Tyler Sizemore and principal investigator Dr. Andrew Dax. Our lab centers around the mechanisms of what is known as neuromodulation. Basically, neuromodulation is the system by which neural networks in animals automatically adjust behavior according to both internal and external stimuli. This is an automatic process, and it basically works by altering the abilities of certain neurons to respond to signals. Now, I want you to think of a time that you were starving. Did you notice just how intoxicating the scent of food was? Well, that was your body using your hunger state to affect your perception of odors. That is neuromodulation in action. That example actually had a lot to do with our work as we study the effect of neuromodulation on olfaction, or the ability to detect odors. I shouldn't have to tell you that the ability to find food is one of the most important things for a living being to have. Well, it just so happens that the sense of smell is one of the most important contributing factors to food finding success. And since neuromodulation is such a huge part of that, of course it's in our best interest as biologists to understand it. To study this in our lab, we use vinegar flies as a model organism, as not only are their nervous systems simpler than a human's, but they are also structurally similar. For our ongoing experiment, we're suppressing a specific serotonin receptor which might play a role in one of the most important types of neurons used in neuromodulation, the local interneurons. These are basically neurons that interpret really small bits of olfactory information. The receptor we're suppressing is known as the 5-HT1A receptor. Given that we know serotonin and its receptors are already there in the local interneurons, it's possible that they play a role in odor sensitivity, and that's what we're investigating. To test the flies that have had their 5-HT1A receptor expression suppressed, we place them in what is known as a T-maze assay, as shown in Figure 1. This is an already established test which involves placing flies at the bottom of a T-shaped arena and then letting them make the choice of moving towards an odor acting as a test stimulus, moving towards water, which is used as a control, or staying in the start area. We tally up the flies based on where they end up, and we interpret that as choices. For our test stimulus, we're using apple cider vinegar, a known attractive odor for vinegar flies. And in this project, we've done so at concentrations of 1 to 10 and 1 to 100. Moving on to Figure 2. In order to actually suppress the expression of the receptors, we make use of a method of studying gene expression known as the UAS-Gal4 system. The way it works is that it uses a Gal4 transcription factor, which in this case is called R32F10, that is basically located in such a way on the genome that it affects a certain type of cell. Then there's the UAS gene, which carries the effector sequence. When UAS and Gal4 are combined, the effect that's tied to UAS specifically affects the targets of the Gal4 factor. So in this case, RNA that specifically knocks down the 5-HT1A receptor expression is introduced so that it only affects local interneurons. In this diagram, what I just described is what the last combination of genotypes results in: UAS, 5-HT1AI, R32F10. All other genotypes are controls, and they exist and are tested so that we can know for sure that an effect we observe isn't due to a confounding variable. Now, we hypothesized that reducing expression would cause some sort of effect, but actually, if you look at the data distribution shown in Figures 3 and 4, you'll see that the performance indexes haven't been significantly different across control and experimental genotypes, meaning there's no observable effect. Now, does that mean that the 5-HT1A receptor doesn't play a role in olfactory behavior? Well, as of now, that might seem true, at least in this isolated case, but there's still a lot to explore. See, these concentrations we're working with, 1 to 10 and 1 to 100, are actually quite high for vinegar flies, and it's very possible that we might be overwhelming our flies with our odor. That's why we must continue our work and do so at lower concentrations of apple cider vinegar. And we're already getting started with that, with plans to carry out experiments at 1 to 250 and 1 to 1000. Even with a reduction in concentration, there might still be no observable effect, and in that case, we may try switching to a different type of behavioral assay, like one that allows us to track behavior directly. Now, in the course of all this, you might have lost sight of why all of this is important. See, if our hypothesis proves to actually be true and we manage to find an observable effect with 5-HT1A receptor knockdown, we will have the information we need to start investigating neural connections within local interneurons, and that might lead to a discovery that can contribute to the understanding of the incredibly important process of neuromodulation. I hope you were able to take away something from this presentation. Thank you for listening.